You have felt it 100 times without naming it. The moment before the first sip of coffee in the morning, when the mug is in your hand and the smell has reached you but your lips have not touched it yet. The day before a trip you've been looking forward to for weeks. The second before someone you love walks through a door after a long time away. There is a state that lives in those moments. You feel a pull towards something worth having, and it often feels sharper and more vivid than the thing itself. There is a molecule behind that pull. It is called dopamine. And the strange thing is that dopamine has nothing to do with pleasure. That is not a simplification or a provocation. It is what decades of careful work on individual neurons has revealed. Dopamine does not tell you how good something feels. It tells you how much something is worth wanting. Those two things look identical from the outside. Both drive you towards something. But they involve completely separate systems in the brain, and only one of those systems is dopamine. The system that handles pleasure is older, quieter, and far less studied than the one that handles the pull. This confusion is not a small misunderstanding. It has shaped how researchers have interpreted addiction for decades. It has shaped how motivation disorders have been framed, how treatments have been designed, and what it means when someone says they can no longer bring themselves to care about the things they used to love. Getting dopamine wrong sends all of those questions in the wrong direction, and the stakes of getting the direction wrong are high. Consider what it means to treat a craving as a pleasure problem. The logic would be to reduce the pleasure available from the substance, or to make the experience aversive enough that the person stops wanting it. But if wanting and pleasure run on separate systems, a person can remain in the grip of intense wanting long after the substance has stopped delivering anything resembling pleasure. They know the experience will disappoint. They have had that experience repeatedly. The wanting does not adjust on the basis of that knowledge. The clinical description follows, not a failure of willpower or a preference for the substance, but a wanting that has become detached from what the experience actually delivers and cannot be corrected through reasoning alone. That is a different problem from what most people assume when they hear the word addiction, and it requires a different account of what the brain is doing. Near the base of your skull, tucked into the midbrain, a small cluster of cells is the source of this signal. A few 100,000 neurons at most, out of the roughly 86 billion the brain contains. You could hold the physical space they occupy between two fingers, but from that origin, long fibers extend throughout almost the entire brain. They reach upward into the frontal regions that handle planning and decision. They reach outward into the circuits that determine what is worth approaching. They reach inward to the ancient structures near the brain's center that drive craving and the pull toward things the brain has learned to pursue. This tiny collection of cells reaches nearly everywhere the brain makes a judgment about what to do next. What they broadcast is not a pleasure signal. It is something more like a continuous calculation, comparing what the brain expected with what it got, and sending out the difference. Understanding that calculation is what changed when researchers finally placed fine electrodes beside individual dopamine neurons and listened to what those neurons were actually saying. The data they collected rewrote what this molecule was understood to be, and it started, as so many advances in neuroscience do, with a wrong assumption that had been held so long it had come to feel like common sense. How does a molecule that has nothing to do with pleasure end up controlling almost everything you pursue? Why does it fire before the reward arrives and go silent the moment it does? And what happens to wanting when the expected thing never comes? If you enjoy exploring questions like this about how the mind works, a like would be highly appreciated. And if something in what follows raises a question or a thought you want to share, the comments are there for exactly that. With that said, let's get back to the topic. To understand what dopamine is doing, you have to start with where it comes from, because the answer to that question is one of the most counterintuitive things about the whole system. A power station the size of a small room sits buried beneath a vast city. Its cables run out to every neighborhood, every street, every building. The city has millions of lights. The station has a handful of workers. That is roughly the geometry of the dopamine system. Two small clusters of cells sit in the midbrain, deep near the base of the brain, each one compact enough that both structures together could rest on the tip of your finger. And from those two clusters, long fibers stretch outward like cables reaching nearly every part of the brain that shapes a human life. They reach what you pursue, what you avoid, and what you feel compelled to return to. The first cluster is called the ventral tegmental area. Researchers use its initials, VTA, because the full name is cumbersome and the letters appear constantly in the literature. The second cluster sits just beside and slightly below it and carries a name that describes its appearance rather than its function, the substantia nigra, meaning the dark substance. Under a microscope, these neurons are visibly darker than the surrounding tissue. A pigment called neuromelanin accumulates as these cells work, staining them dark enough to be recognizable at a glance. Early anatomists named the structure before anyone had any idea what it produced. The discovery of what these darkened cells were actually doing came more than a century after they were first described. In the late 1950s, a Swedish pharmacologist named Arvid Carlsson was working on a class of brain chemicals called monoamines. At the time, dopamine was understood only as a stepping stone, a precursor on the chemical pathway toward other signaling molecules. It was not considered a messenger in its own right. Carlsson doubted this. He gave laboratory animals a drug that depleted the brain of its monoamine stores. The animals became rigid and unable to initiate movement, resembling the symptoms of what would later be understood as Parkinson's disease. He then administered L-DOPA, the chemical precursor the brain converts into dopamine, and the animals recovered. When he measured dopamine levels across different brain regions, he found it concentrated separately from the other monoamines, in regions involved in movement and motivation. His conclusion was straightforward. Dopamine was a neurotransmitter in its own right, not a stepping stone, a signal with its own destinations and its own purpose. The work was not immediately accepted. It took more than 4 decades for Carlsson to receive the Nobel Prize in Physiology or Medicine in the year 2000 for a finding his colleagues had once regarded as unlikely. By then the field had confirmed what he suspected. Those two small clusters in the midbrain were not incidental structures. They were something close to the engine of the brain's motivational life. What makes the anatomy so striking is the disproportion between their size and their reach. A single axon from one of these cells can branch hundreds of times, touching thousands of neurons across enormous distances. One cell in the VTA can have terminals in the nucleus accumbens, the prefrontal cortex, and the hippocampus simultaneously. One burst of firing sends a signal into all of those regions at once within milliseconds. The anatomy functions more like a broadcast tower than a telephone line. A single message goes out. It arrives everywhere the branches reach at the same moment. The substantia nigra projects differently. Its fibers run densely into the striatum, the structure involved in initiating and smoothing voluntary movement. This became unmistakably clear through disease. When the pigmented neurons of the substantia nigra begin to die, movement loses its fluency. A tremor appears at rest. Starting an action becomes effortful in a way it never was before. Arvid Carlsson's successor, Ole Hornikiewicz, examined post-mortem brains from Parkinson's patients in the 1960s and found the striatum almost completely depleted of dopamine. The cells that had been quietly supplying it were gone. Their absence revealed exactly what they had been providing. The signal that releases movement from the competing pressures holding it in check had gone silent, and the body could no longer begin what the mind intended. The two pathways have come to be understood as handling separate aspects of the dopamine system's job. The pathway from the substantia nigra to the striatum handles the initiation and fluency of movement. The pathway from the VTA runs toward the structures involved in motivation and learning, most prominently the nucleus accumbens and the prefrontal cortex. In practice, these pathways interact in ways that make clean separation difficult, but the disease states that arise when each is damaged separately have been the clearest window into what each one is providing. Movement disorders reveal one pathway's function by removing it. Motivation and addiction research reveals the other. The signal originates in a structure the size of a thumbnail. Its effects span nearly the entire brain. A story settled into the scientific literature about dopamine in the years before anyone had the tools to test it properly. The story went like this. Something good happens and the brain releases dopamine. It rises at the approach of pleasure and falls when pleasure leaves. The cells that produce it are the brain's pleasure centers. They fire when rewards arrive and teach the rest of the brain to seek more of the same. That story felt coherent. It matched what anyone could observe from the outside, and it was built on an experiment from the 1950s that seemed, at the time, to close the question entirely. James Olds was a postdoctoral researcher at McGill University in Montreal when he made the observation that would define his career. He and his colleague Peter Milner were implanting small electrodes in rats and allowing the animals to trigger brief pulses of electrical stimulation by pressing a lever. The experiment was designed to study something else entirely. One rat pressed the lever with a frequency that no one expected. It pressed again and again, returning hundreds of times an hour, ignoring food and water until it was physically exhausted. When Olds and Milner mapped the electrode position, they found it placed near a pathway that would later be understood to include dopamine-projecting fibers. The rat was working harder for this stimulation than for almost anything else in its experience. It would cross an electrified floor to reach the lever. It would choose stimulation over sleep. Olds named the region a pleasure center, and the name spread quickly through the field, carried by the sheer force of how clearly the rat appeared to prefer stimulation to almost everything else that might sustain its life. Later experiments on human patients with electrodes implanted for medical reasons seemed to reinforce the conclusion. Some patients, when certain circuits were stimulated, described a feeling of pleasant anticipation and asked for the stimulation to be repeated. Their descriptions were often vague and difficult to characterize precisely. Some found it difficult to name the experience at all, but the request for repetition appeared to confirm the fundamental idea. This pathway was something the brain wanted more of. The assumption grew that stimulating dopamine circuits was equivalent to producing pleasure. When researchers later found that cocaine and amphetamines sharply elevated dopamine in the nucleus accumbens, the region at the end of the major motivational pathway, the framework seemed complete. The drugs made animals work just as frantically as the lever had. They would press for cocaine or amphetamine with the same relentless urgency the lever rat had shown. The common variable was dopamine. If dopamine drove the behavior, and the behavior was pleasure-seeking, then dopamine must encode pleasure. The assumption felt like an explanation. It was taught in textbooks. It shaped the language researchers used to describe what they were studying. But there was a question buried in this reasoning that none of these experiments could answer. The rat pressed the lever frantically. Was it pressing because the stimulation felt good? Or was it pressing because the stimulation created a powerful pull toward pressing, entirely independent of whether the experience was pleasurable at all? From the outside, these two possibilities look identical. Both produce an animal pressing as though its life depends on it. The difference is entirely internal, and the rat could not report it. The question mattered because the two answers lead in completely different directions. If the signal is pleasure, then pleasure is what drives behavior and what goes wrong in addiction. If the signal is the pull toward things, then wanting is a separate process from enjoying, and a system can be trapped in the pull without any corresponding satisfaction at the end of it. The lever rat became the foundation of a framework without anyone ever knowing which answer was correct. The tools needed to answer that question did not yet exist. Distinguishing wanting from liking required measuring something other than behavior. It required listening to the dopamine neurons themselves, one at a time, while an animal was still engaged with the world. That technical ability was being developed in the late 1980s in a laboratory in Switzerland. Wolfram Schultz was a neuroscientist working at the University of Fribourg in Switzerland when his laboratory developed the technical ability to record electrical signals from individual dopamine neurons in the brains of awake, behaving monkeys. This was not a trivial accomplishment. A fine electrode had to be placed with enough precision to pick up the signal from a single cell without disrupting the animal or the behavior being observed. The electrode had to remain stable while the monkey moved and responded to its environment, and the recordings had to be interpreted cleanly enough to separate one cell's signal from the background noise of thousands nearby. Once it worked, the recordings produced data that no one had seen before at that resolution. The initial setup was straightforward. A monkey would sit in a chair, and at an unpredictable moment, a small amount of fruit juice would be delivered to its mouth. Schultz's electrode sat near neurons in the ventral tegmental area, listening to what those neurons said when the juice arrived. The neurons fired. When the juice was delivered unexpectedly, dopamine neurons produced a sharp burst of activity. This appeared to confirm exactly what the field had assumed. Something good happened, and dopamine fired in response. The pleasure signal, apparently right on schedule. Then the experiment changed. A light was added, flashing a moment before the juice arrived. After several repetitions, the monkey learned to expect the juice when the light appeared. Schultz kept watching the neurons, and the firing pattern shifted. The dopamine neurons no longer responded when the juice arrived. They responded when the light appeared. The moment of the reward, now entirely predictable, produced nothing from these cells. The moment of the cue, which predicted the reward, produced the burst of activity that the reward itself used to generate. The firing had moved backward in time. It had moved to the moment of prediction. This was striking enough on its own. But the decisive observation came when the light flashed and the juice failed to arrive. The monkey waited. The expected moment of delivery came and went. And at precisely the moment when the juice should have been there, the dopamine neurons dipped below their resting rate. Their activity fell below baseline. The absence of the expected reward was not neutral. It produced an active signal running in the opposite direction. What Schultz was seeing was a three-state system. When something arrived that was better than predicted, the neurons fired above baseline. When something arrived exactly as predicted, they held flat. When something expected failed to appear, they fell below baseline. The signal was not reporting the presence of a reward. It was reporting the relationship between what the brain had predicted and what actually arrived. Schultz and his team also tested what happened when an unexpected, unpleasant stimulus arrived instead of a reward. The dopamine neurons did not fire, they paused. The same cells that burst when something good arrived unexpectedly went quiet when something bad arrived unexpectedly. The signal had a positive arm and a negative arm, each tracking the same underlying calculation from a different direction. This was not a pleasure signal being hijacked by bad news. It was a prediction error signal, and errors can run in both directions. The formal paper describing these findings, published in Science in 1997, became one of the most cited papers in neuroscience of the following decade. But the finding had been accumulating across years of careful recording, and its implications for everything the field had assumed about dopamine were substantial. The molecule that everyone called the pleasure chemical was not encoding pleasure. It was encoding surprise. It was registering the distance between what the brain had expected and what the world had actually delivered, and broadcasting that distance through the projection pathways so that the brain could update its model. That shift in understanding had a parallel in a field far removed from neuroscience. Computer scientists working on reinforcement learning had been building algorithms that updated the value of expected outcomes based on exactly this kind of error signal. They called it temporal difference learning. What Schultz had found in living tissue looked like the biological implementation of the same principle. The brain had arrived at the same solution that engineers were formalizing in code. The gap between expectation and reality was being computed at the cellular level and broadcast as an update signal. The learning function appeared to be primary, with what feels like pleasure from the inside being what the system produces when the learning signal fires in a strongly positive direction. What Schultz had uncovered was not just a curiosity about monkey brains responding to juice. It was a description of a principle the brain uses continuously to learn about the world. The signal is called a reward prediction error. The name is precise. It is the difference between the reward the brain predicted and the reward that actually arrived. When reality exceeds prediction, the error is positive and dopamine rises. When reality matches prediction exactly, the error is zero and dopamine stays flat. When reality falls short of prediction, the error is negative and dopamine dips below its resting rate. Think of a spirit level, the tool a builder uses to check whether a surface is flat. The bubble sits in the center when the surface is perfectly level. The moment the surface tilts, the bubble moves to show how far and in which direction. The level does not measure how flat something is, it measures deviation from flat. A perfectly flat surface produces no signal at all. Dopamine works by the same principle. The neurons are not measuring how good something is in any absolute sense. They're measuring how much better or worse it is than the brain predicted. A wonderful outcome that was fully expected produces no spike. A modest outcome that was completely unexpected produces a large one. The signal is always about the gap, never about the absolute value. This reframes something that most people have noticed but never fully explained. Think of a time when you anticipated something for weeks, imagined it in detail, and then found it somehow flat when it arrived. The thing was good. It matched what you hoped for. But the feeling was not there in the way you expected. That is not disappointment, and it is not ingratitude. It is the prediction error collapsing to zero because the brain had already incorporated the outcome into its model. The signal had already been spent in anticipation. The arrival confirmed a prediction that was already accurate, and an accurate prediction generates nothing. The reverse is equally true. An unexpected piece of good news, arriving with no warning, can carry more force than something much larger that you spent months anticipating. The gap was still open. The signal fired. The brain was caught without a prediction to absorb the difference, and the full magnitude of the error registered in a way that no amount of long-anticipated good news can replicate. And here is the thing that most accounts of dopamine leave unaddressed. If the dopamine spike is already gone by the time the predicted reward arrives, what carries the satisfaction when the thing actually comes? If dopamine is not running when you finally get what you wanted, where does the pleasure come from, and what generates it? The reward prediction error framework answers what dopamine is doing. It raises a new question about what the arrival experience belongs to. Those two things are separated by more than a theoretical distinction. The conditions under which that separation becomes clearly visible turned out to exist in the laboratory, and the experiment that revealed it had implications that extended far beyond the prediction error account. The prediction error framework also explains something about how new places feel different from familiar ones. When you visit somewhere you have never been, the brain does not know what to expect. Every sound is unclassified. Every corner holds information the model has not yet processed. Every moment carries a mild positive prediction error because novelty signals potential significance that has not yet been incorporated into the model. The world feels more vivid. Time seems to expand slightly. The dopamine system is logging new information, not confirming old predictions. This is also why novelty seeking has a neurological basis rather than being purely a matter of personality. The brain is shaped to weight new information heavily, because in an uncertain environment a new signal could indicate danger or opportunity. The cost of ignoring a new signal that turns out to matter is higher than the cost of attending to one that turns out to be irrelevant. The system defaults to treating novelty as significant. The vividness you feel in a new environment is not an aesthetic response to change. It is the brain's prediction machinery running at a higher rate than it does in familiar territory, assigning temporary significance to inputs it has not yet classified. Once those inputs are classified, the machinery quiets. The novelty was not in the environment. It was in the gap between what the brain predicted and what it encountered. As a place becomes familiar, those prediction errors close one by one. The brain knows what to expect. The signals arrive as predicted. The dopamine response quiets, not because the place has become less beautiful, but because the model is accurate. The liking may still be there, carried by a different system entirely. But the component of experience that felt like intensity, the part driven by prediction error, is no longer firing. This is not loss, it is learning. The brain has done its job. The quieting is the mark of a model that has grown more precise. But it does mean that the feeling of intensity is not a property of the thing being experienced. It is a property of the gap between what was expected and what arrived. Close that gap completely, and the intensity goes with it. The Schultz recordings also revealed something about what happens to the signal as that gap closes over time. It was not just that dopamine neurons fired when rewards arrived. It was what happened to that firing across repeated exposures as the animal learned. Early in training, when the reward was new and completely unpredicted, the dopamine spike landed at the moment of delivery. But as learning proceeded, something shifted in the timing. The spike moved. It migrated backward from the moment of reward to the earliest reliable signal that predicted it. By the end of training, the light that preceded the juice was producing a full spike. The juice itself, arriving exactly when expected, produced nothing. This migration happens through a precise mechanism. Each time a cue reliably precedes a reward, the brain updates the value of that cue, and the prediction error shifts one step earlier in the sequence. After enough repetitions, the error has moved all the way back to the cue. There is no gap left to close at the moment of delivery. The prediction was already accurate. Think of someone who has made tea at the same time every morning for years. Early on, the pleasure of the first sip was what drove the routine. But after 1,000 mornings with the same kettle, the sound of the water coming to the boil is already enough. The body orients toward the mug before the tea is poured, drawn by the expectation that has been confirmed so many times it no longer needs the outcome to fire. The response has migrated to the earlier signal in the sequence. That is what the dopamine neurons are doing at the level of the cell. The response is not slower or weaker after learning. It is sharper and more precise, and it fires at the first reliable point in the sequence rather than the last. The brain has learned to predict, and the signal marks that the sequence is still running true. The signal does not just move to the cue closest in time to the reward, it moves to the earliest reliable predictor in the whole sequence. If a sound reliably precedes a light that reliably precedes a reward, and the sequence is learned thoroughly enough, the dopamine spike migrates all the way back to the sound. Each element becomes a cue for what follows, and the prediction error credit shifts backward to the most informative point the brain can access. This is exactly what the computational models predicted. The signal seeks the moment that lets the brain begin updating its expectations as early as possible. The direction of that movement reveals something about how the brain assigns credit across a sequence. The credit runs backward through time rather than forward. This means the signal is not evaluating what just happened. It is evaluating what, in retrospect, was most predictive of the outcome. Every new experience is being mapped backward through the events that preceded it. The earliest link in a reliable sequence becomes the point at which the brain most strongly marks the value of what follows. This is how a brand name or a physical setting can acquire the motivational charge that the product itself originally carried. The charge does not belong to the product. It belongs to the earliest reliable signal in the sequence the brain learned to follow. This process is part of what makes habits so durable and advertising so effective. Early in a routine, the reward at the end drives the behavior. Over time, the cues at the beginning of the routine carry more and more of the motivational load. The wanting fires at the first signal in the sequence, before any of the actual substance of the routine has begun. A brand, a jingle, a familiar package can all migrate the dopamine response forward of the actual product. The wanting fires at the image, not at the thing. The brain is doing exactly what it was shaped to do: respond to the earliest reliable signal that something worth pursuing might be nearby. The consequence of this migration is subtle, but important. Once a reward is fully predicted, it generates no dopamine spike. A habitual pleasure, perfectly timed and exactly as expected, arrives in silence from the dopamine system's perspective. There is no error to signal. The model is accurate. The brain simply confirms what it already knew, and this is where the separation between wanting and something else becomes impossible to ignore. If the dopamine spike fires at the cue and not at the reward, what is actually happening at the moment the reward arrives? What does the brain experience when the predicted thing finally comes, if dopamine is not the molecule producing that feeling? The answer to that question required a different kind of experiment entirely, one that removed dopamine from the equation and watched what remained. In the mid-1980s, Kent Berridge was working at the University of Michigan on a puzzle that seemed at first like a confirmation of what everyone already believed. The standard account held that dopamine was the brain's pleasure signal. When dopamine flowed, pleasure followed. The wanting and the liking were thought to be one system, powered by one molecule. Berridge's experiments produced a result that overturned that account entirely. Berridge and his colleagues used a technique that depleted the dopamine in rats almost completely. They were meticulous. The animals were left with around 1% of normal dopamine levels in the relevant pathways. By the standard account, these rats should have lost the capacity to experience pleasure. They did not want to eat. Left to their own devices, they stopped pursuing food and would eventually starve without intervention. The wanting was gone. Then Berridge placed a drop of sugar solution directly into the mouth of a depleted rat. The animal could not go and find the sugar. It had no motivation to seek it out. But when the sugar arrived at the tongue, the rat's face responded. A particular pattern of tongue movement and expression appeared, the same pattern seen in normal rats and in human infants when something sweet arrives unexpectedly. The response was brief, localized, and unmistakable. The rat was experiencing something. The pleasure reaction was intact. Wanting had been removed. Liking remained. Berridge ran the experiment in reverse as well. He found ways to amplify dopamine activity in the nucleus accumbens without affecting the other signaling systems involved in the hedonic response. Rats in this condition became intensely motivated. They pursued food with urgency and worked harder for every reward. When the food arrived, the hedonic facial reaction was normal, unchanged from baseline. The wanting had been turned up. The liking had not moved. This was the first clean separation of two things that had always been assumed to travel together. The dopamine system drives the pursuit. A different system entirely handles the arrival. Berridge identified the second system more precisely over subsequent years. It runs primarily on opioids rather than dopamine, and it is concentrated in specific small clusters of cells within the nucleus accumbens shell. Blocking the opioid signal in those clusters reduced the pleasure reaction to sweetness even when dopamine was fully intact. Berridge called these clusters hedonic hotspots. They are very small. In the rat, the primary hotspot occupies a region that can be measured in millimeters. The structure that generates the felt experience of pleasure when directly manipulated is anatomically far smaller than anyone expected. This means the standard account of the reward system as a single region generating a unified response is wrong. The nucleus accumbens handles both systems, but in different parts and through different mechanisms. The larger dopamine-driven portions generate the wanting signal, the pull toward things the brain has learned to pursue. The small opioid-sensitive clusters generate the liking response when the pursued thing finally arrives. Two systems sharing the same structure, running on different molecules, producing fundamentally different experiences. They appear unified only because they happen to be close enough in the brain that no one noticed the separation for most of the 20th century. Berridge coined the term incentive salience to name what dopamine actually does. Salience means the quality of standing out, of drawing attention. Incentive salience is the specific quality of standing out as worth pursuing. Dopamine does not label a thing as pleasant, it labels a thing as worth moving toward. And the separation Berridge documented in rats holds in human neuroscience as well, confirmed through imaging and lesion studies across the decades since. The clean separation Berridge documented also clarifies something about the language researchers and clinicians use when they talk about reward. When someone says they can no longer feel pleasure in things they used to love, two different conditions can produce that description from the outside. In one, the liking system is diminished and the experience of enjoyment is genuinely blunted. In the other, the wanting is intact or even elevated while the arrival experience fails to match it. Both present as a loss of pleasure. Both require different explanations and different approaches to address. Berridge's framework gave researchers a way to ask which system was affected, rather than treating pleasure as a single, undifferentiated thing that was simply present or absent. Dopamine does not label a thing as pleasant. It labels a thing as worth moving toward. That distinction sounds fine as a definition. Lived from the inside, it has a specific texture that most people have experienced without having a name for it. When you feel the pull toward something, what is actually happening is that the dopamine signal has attached incentive salience to that object or cue. The wanting fires. What that wanting is predicting is an arrival experience that will satisfy it, but the prediction is about liking. The mechanism doing the predicting is not the liking system, and when the arrival comes, the liking system produces an experience calibrated to the actual value of the thing, not the intensity of the wanting that preceded it. This is why anticipation so often outstrips satisfaction. You have probably noticed this without having a word for it. An anticipated thing rarely feels as good as the anticipation felt. A destination reached rarely matches the charge of the journey toward it. The wanting was intense. The liking when it came was real but quieter. The wanting arrives first and projects forward. The liking arrives after and registers only what is here. The two are not operating on the same time horizon. A large wanting does not guarantee a large liking. Wanting is about prediction, and liking is about reality, and predictions routinely diverge from what arrives. Incentive salience is also not fixed. The same object can pull with very different force depending on the state the brain is in when the cue appears. Hunger amplifies the incentive salience of food cues. Stress can amplify the pull toward cues associated with relief. The intensity of wanting is not a stable property of the thing being wanted. It is the product of the cue and the internal state of the system that assigns meaning to it. This is part of why the same food or the same habit can feel irresistible in one moment and entirely manageable in another, with no change in the external situation. This state dependence has another important feature. The incentive salient signal does not extinguish easily. Once a cue has been associated with a reward and charged with dopamine-driven significance, that charge tends to persist. Think of checking a mailbox every day after switching to email entirely. The pull toward the mailbox at the familiar hour does not immediately stop. The cue is still in the environment. The time of day still triggers the sequence. The wanting fires because the association was learned, and learning runs in both directions, fast to acquire, slow to release. The signal does not read the current situation and decide whether the pull is still warranted. It fires on pattern recognition, not on a reassessment of relevance. This is part of what Berridge meant by incentive salience being separable from hedonic value. The wanting is not a measure of how much the thing will be enjoyed. It is a measure of how much the brain predicts it will be enjoyed, calibrated by past experience and current state. And past experience, once encoded, does not update automatically when circumstances change. The cue continues to pull even when the person knows, consciously and clearly, that the arrival will not be worth the pull. The knowing lives in one system. The wanting lives in another. Berridge's framing also helps explain something that most accounts of pleasure leave unaddressed. Why even when the liking arrives and is genuine, the wanting resumes so quickly. The liking is a response to what came. The wanting is already reaching for what comes next. The liking registers what is already here, and what is already here is always, by definition, already passing. The wanting project forward into the next moment. The loop does not stop because the thing arrived. It stops only when the cue disappears or the state changes. There is also something worth understanding about why the wanting signal is calibrated to run continuously, rather than stopping when satisfaction arrives. From an evolutionary perspective, a system that stops generating incentive salience the moment a reward is collected would be poorly suited to environments where the next reward is never guaranteed. The wanting that resumes almost immediately after a satisfying meal or a moment of genuine rest is not a sign of insatiability. It is the system doing its primary job, keeping the organism in motion toward the next thing before the current situation deteriorates. Wanting that stops cleanly and permanently at satisfaction would be a system oriented entirely toward the past. The one that evolved orients toward what comes next. When wanting and liking travel together, as they do under ordinary circumstances, the system works as it was built to work. You see something worth pursuing, and the wanting signal fires. You move toward it. It arrives, and the liking registers. The gap between what you predicted and what you got closes. The prediction error confirms that the world behaved as expected. The two signals form a loop, and the loop corrects itself after each cycle. The loop breaks when wanting and liking are forced apart, and the clearest way to force them apart to make the separation sudden and visible is to flood the wanting system far beyond anything the liking system can match. Drugs that act on the dopamine system do not work by tricking the brain into thinking something pleasurable happened. They work by bypassing the mechanism that would normally require a real-world outcome to justify the signal. Cocaine blocks the transporter that clears dopamine from the synapse, so dopamine accumulates far beyond its normal concentration. Amphetamines go further. They reverse the transporter entirely, forcing dopamine out of the neuron whether or not the cell has been activated. The result is not a signal saying something good happened. It is a prediction error of enormous magnitude, reporting that something unimaginably good is occurring with no real-world event to attach it to. The wanting fires without the liking anywhere nearby. Over time, the system pushes back. The dopamine neurons themselves detect the excess through receptors on their own surface and reduce their output. Receptor populations on the downstream cells shrink, because the cells are receiving a signal so far above any normal range that reducing their sensitivity is the only way to keep functioning. The system recalibrates around a new, much higher baseline. Natural rewards now fall short of it. A good meal, a piece of music, a conversation that once felt sustaining no longer clears the threshold. The prediction error for ordinary pleasure turns negative. The signal says not enough, expected more. This is the mechanism behind tolerance, and it runs in two directions simultaneously. The liking system becomes less responsive as its hedonic hotspots are exposed repeatedly to opioid activity at levels they were not shaped to sustain. The wanting system becomes more reactive because cue associations are being strengthened every time the substance is encountered. The wanting grows, the liking contracts, the gap between them, which the system was designed to close at every cycle, stays open. The consequence of this recalibration is visible in withdrawal. When the substance is removed, the brain is operating with a baseline that was set by dopamine concentrations far higher than any natural experience can produce. Natural rewards do not just feel less satisfying. They register as mildly negative, because the brain's model has been calibrated to expect more than they can deliver. The physical discomfort of withdrawal is in part the system attempting to return to a set point it no longer has. The dopamine neurons that reduce their output under chronic excess are slow to recover their baseline firing rates. The receptors that contracted their numbers under prolonged signal are slow to rebuild. The body is in a state where the model expects a signal level that is no longer arriving, and every moment of ordinary experience is measured against that expectation and found short. What does not recalibrate at the same rate is the pull. The incentive salience mechanism, the dopamine-driven response to cues associated with reward, remains sensitive even as the ability to experience liking at arrival declines. Cues that were associated with the substance during earlier use retain their charge. A smell, a location, or a particular emotional state present during earlier use can all produce a dopamine response that the absence of the substance has not erased. The cue fires the wanting. The wanting does not check whether the liking will be proportionate. A person who has been through repeated cycles of this shift may know clearly, at a conscious level, that the thing will not deliver what it once did. That knowledge is real, and the pull toward the cue is unchanged, because knowing and wanting run on different systems, and the wanting system does not update on the basis of declarative knowledge. It updates on the basis of prediction error, and the loop has been running in a way that no natural experience can correct. Berridge called this state wanting without liking, and described it as one of the clearest demonstrations of what incentive salience actually is. The wanting is real. The pull is genuine. It is not a confused preference or a failure of reflection. It is the dopamine system doing exactly what it was built to do, following cues and firing the signal that says move toward this. The system simply cannot read the verdict that experience has already returned, and so the loop does not close, it runs again. Knowing and wanting run on different systems. That sentence sounds like it should have obvious implications, but following those implications leads somewhere most people have not fully thought through. The prefrontal cortex handles planning, judgment, and the evaluation of future consequences. It receives dopamine input and plays a central role in deliberate reasoning. When you recognize that the pull you're feeling is disproportionate to what the arrival will actually deliver, that recognition is being generated there. The assessment is real and it is correct. But the prefrontal cortex does not cancel the dopamine signal in the nucleus accumbens. It can inhibit the behavior that the signal is driving. It can hold back the action. It cannot stop the wanting from being generated in the first place. The signal fires. The cortex catches it and suppresses the response. The signal fires again. The cortex suppresses again. This is not the same as the wanting not being there. It is the wanting being there and being held, which requires continuous effort in a way that automatic behavior does not. This asymmetry matters for understanding why sustained wanting is so draining. Holding behavior in check against a persistent motivational signal draws on the same prefrontal resources used for planning, attention, and decision-making. Those resources are not unlimited. Under fatigue and stress, the prefrontal cortex becomes less effective at maintaining inhibition. The wanting does not increase in those moments. The braking decreases. The wanting was always there. The braking was what changed. The speed difference between the two systems adds another layer. The dopamine signal reaches the nucleus accumbens and the limbic structures quickly, propagating through pathways that evolved long before the prefrontal cortex reached its current size. The prefrontal evaluation arrives later, after the initial motivational burst has already landed. In any encounter with a strong cue, the wanting fires first. The assessment of whether to act on it comes after. The sequence is not parallel, it is sequential, and the wanting has the head start. This asymmetry has an origin that predates the prefrontal cortex by a long margin. The structures that generate the dopamine signal evolved in animals that had no frontal evaluation capacity at all. The wanting system is built on machinery that was already ancient when the capacity for deliberate reasoning began to develop in primate lineages. The two systems were not designed in parallel. The cortical capacity was added on top of an existing motivational architecture. The result is that the evaluation always arrives after the signal it is meant to evaluate. In low-stakes situations with weak cues, the delay is unnoticeable. In high-stakes situations with strong cues, the delay is the interval in which most decisions that people later regret are made. The wanting fires. A moment later, the reasoning arrives. In that moment, behavior is already in motion. This also clarifies something about the states in which wanting is hardest to manage. Poor sleep, high stress, and emotional depletion all reduce prefrontal function. They do not create new wanting. They reduce the braking. The pull was there all along. The conditions that make it feel overwhelming are the conditions in which the system meant to hold it back is least available. The wanting feels as though it grew. What actually happened is that the resistance thinned. The signal was unchanged. The brake was what failed. There is one more feature of the wanting system that matters here. The incentive salient signal does not update based on verbal knowledge. Telling yourself that the pull is disproportionate and that the arrival will disappoint does not recalibrate the signal. The dopamine system updates on prediction error, not on reasoning. It requires actual experience of an outcome that was worse than predicted to begin eroding the learned association. And even then, the erasure is slow. Extinction, in learning terms, is not the deletion of an old association. It is the formation of a new one alongside it, a new memory that the cue no longer reliably predicts reward. But the old association remains encoded. Under stress or fatigue, in a context that resembles the original acquisition, the older association can resurface. The wanting that seemed to have quieted can return with most of its original force. This is not a theoretical vulnerability. It is visible in clinical outcomes. A person who has been abstinent for months or years can encounter a familiar cue and find the wanting, returning at something close to its original intensity, even though the association has been inactive for the entire interval. The old encoding was not erased. it was suppressed by a competing association formed during abstinence. Under the right conditions, the suppression lifts and what was formed first reasserts. The brain holds two records of the same cue. The more recent one predicts that nothing will follow. The older one predicts something very different. Which record runs depends on the state the brain is in at the moment the cue arrives. Pull everything back to a single mechanism and it becomes visible. Dopamine is not, at its root, a pleasure chemical, or a movement chemical, or a craving chemical. It is the signal the brain uses to compare what it predicted with what actually happened, and to update its predictions accordingly. The brain is not a passive observer of the world. It runs continuously forward, generating models of what is about to happen, and measuring every arrival against its current best guess. Every sensation that comes in is compared to an expectation. Every movement begins with a prediction of what that movement will feel like. Every reward outcome is checked against what the brain calculated it would be. This comparison runs in every sensory system, in every moment of motor planning, in every assessment of value. It is not a feature of one region. It is the fundamental operation of the brain. Dopamine is the currency of that comparison. A positive prediction error says the model was wrong in the good direction and should update upward. A negative prediction error says the model was wrong in the bad direction and should update downward. A zero error says the model was accurate and no update is needed. Over thousands of repetitions, these updates build and refine the brain's working model of the world into something that can make better and better predictions. In the mid-1990s, Peter Dayan and Red Montague worked with Terence Sanowski to show formally that the behavior of dopamine neurons was mathematically identical to a class of algorithms called temporal difference methods. These algorithms had been developed in computer science for training systems to make decisions in uncertain environments. They worked by comparing the predicted value of each state with what actually arrived, and adjusting the prediction at each step rather than waiting for a final outcome. The agent did not wait until the end of a sequence to evaluate what happened. It updated continuously, assigning credit backward through time to the cues and states that predicted the eventual result. The algorithm was designed for agents navigating environments where outcomes are delayed and uncertain. It has to estimate the value of each intermediate state and update those estimates as new information arrives. The brain faces the same problem in every domain where consequences are not immediate. The temporal difference framework addressed this by computing partial credit at each step, rather than assigning credit only when a final outcome is reached. The brain had arrived at the same solution through evolution. The details differed. The computational principle was the same. This recontextualizes the two things we know go wrong when the dopamine system is disturbed. When the nigrostriatal pathway degenerates, as it does in Parkinson's disease, the brain loses the prediction error signal that allows smooth motor learning and real-time motor correction. Movement loses its fluency, not only because the start signal is missing, but because the continuous small corrections that keep motion precise are no longer being updated. When the mesolimbic pathway is overwhelmed by substances that flood the synapse far beyond the range the system was shaped to handle, the brain's prediction model for reward becomes radically miscalibrated. The system is not broken in the sense of being unable to run. It is running with a corrupted model, generating predictions that no natural experience can confirm, and the wanting continues in service of a prediction that can never be satisfied. The misconception this episode started with, the idea that dopamine is simply a pleasure chemical, makes a particular kind of sense in retrospect. Under normal conditions, wanting and liking travel together. The prediction error fires at the cue, and by the time the reward arrives, the liking response confirms that the wanting was pointing in the right direction. The two signals are so consistently paired in ordinary life that they appear to be the same signal. The separation only becomes visible when the system is studied directly, either by removing dopamine and watching what liking does without it, or by flooding dopamine and watching what wanting does without liking nearby. What dopamine is actually doing in every moment is answering one question. Was the world better or worse than you expected? That question guides learning and drives motivation. It initiates movement and tells the brain which of its models to keep and which to revise. When it goes wrong, it can trap the system in a loop of wanting that outpaces every available liking by a margin that no arrival can close. When it is working, it is the quiet engine behind every decision about what is worth going toward. Think of the moment again. Something you wanted, almost within reach, not yet arrived. The time before it came carried more charge than the moment it did. You understand now what that charge was. Your dopamine system was running a prediction, and the gap was still open. The cue was in the environment and the sequence was underway. The neurons were firing at the signal that said it is coming, and the calculation says it will be worth going toward. The moment it arrived, if it arrived exactly as expected, the signal collapsed. Not because the thing was disappointing, because the brain had already absorbed the outcome. The prediction was satisfied. The error was zero. There was nothing left to mark. That experience, the charge before and the quiet after, is not a flaw in how you are built. It is the system operating correctly. The brain is designed to want things before it has them, because wanting is what gets you across the distance between where you are and where the reward is. Liking, the quieter signal, is the brief confirmation that the prediction was worth making. The wanting is the engine, the liking is the gauge. The gap between them is also why the moment before often stays in the memory more vividly than the moment of arrival. Prediction errors register strongly because they carry information the brain needs to update its model. A zero error at the moment of a fully expected arrival registers almost nothing. The anticipation was full of open gaps. The arrival closed them all at once and went quiet. Memory encodes the high signal moments, and the high signal moments were the ones before. For most of human history, the gap between wanting and liking was small enough that the loop was self-correcting. The rewards available in the natural world were modest and required genuine effort and uncertainty to reach. The prediction error signal had room to breathe. The brain could reset between cycles, refine its model and move on. The wants were proportionate to what the world could deliver. The liking, when it came, was enough to close the loop and redirect the wanting towards something new. This also explains why the system does not evaluate the long-term consequences of what it pursues. The wanting that reaches toward a meal and the wanting that reaches toward a habit that is slowly undermining a life are running on the same machinery. The same neurons are firing, the same prediction signal is being broadcast through the same pathways. The motivational system did not evolve to assess long-term consequences. It evolved to identify signals that reliably predicted reward in the past and to generate the pull toward repeating them. The capacity to evaluate long-term consequences was added later by a different system, and it arrived with the same timing disadvantage it still carries. Wanting is ancient. Judgment is more recent. And in any contest between the two, the older system carries the structural advantage. The world that a few 100,000 cells were shaped to navigate no longer fully exists. The signals available now can reach magnitudes those cells were never exposed to across the millions of years in which they were formed. When the signal is pushed beyond the range that the wanting-liking loop was built to handle, the system does not simply amplify pleasure. The wanting grows, the liking recedes, and the gap between them, which was meant to be closed by arrival, stays open. But on an ordinary evening, in a system that is mostly working as it should, the signal is simply a calculation running in the background. It tracks the distance between expectation and reality, and pulls you toward what it has learned to predict is worth the distance. It fires at the smell of coffee before the first sip. It fires in the day before the trip. It fires in the second before the door opens. It is there in every moment of reaching, every pull towards something the brain has decided is worth moving toward. Inside your skull right now, a few 100,000 cells are doing this continuously. They are updating their model of the world based on everything that has happened today and everything that has not. They are deciding, in a language below language, what the next moment is worth going toward. The feeling that something is about to be good is not a wish, it is a calculation, and the calculation has been running, without a pause, since the first morning you ever opened your eyes.